So I just want to tell everybody, so I'm a postdoctoral fellow at the Translational Genomics Research Institute, and I came here about 4.5 years ago from the University of Toronto. And my background was actually in brain tumor research, and I didn't know very much about genomics, and only started to get into the world of genomics towards the very end of my PhD. And so one of the main reasons why I moved to TGen was to embark on this exciting era of genomics, and and that's exactly what I'm doing. So today I'm going to talk to you about epigenetics research in myeloma, and I feel this maybe, I don't know how technical I'm going to make it. So we'll just go on. If you have any questions, actually don't be shy. Just ask me, stop me. Okay. So let me just get into what epigenetics is. It's a very hot new area in myeloma. In fact, the MMRF is all over epigenetics research in myeloma, and I'll get into that in just a sec. So we know about the genetic contributions to the disease. Genetic contributions involve generally structural changes to DNA. So you have mutations, you can have copy number changes. And we understand a lot about the genetics of myeloma, and with that information, new treatments have been catered and developed with that. But it's not the only answer. There are other things that can also alter disease, and those are the epigenetic contributions of disease. These are also heritable changes, but they do not involve structural modifications. They generally involve chemical modifications. So there are two main types of epigenetic alterations. One is DNA methylation and histone modification, and the next slide might help a little bit. So epigenomics literally means on top of the DNA. So you have to think about it. DNA is not a naked molecule, okay? It's decorated and dressed with a lot of chemical modifications that alter its structure and therefore function. And one of the types of modifications I'm going to talk to you about, one of the types of modifications involves a small chemical modification that actually attaches to a very specific unit of DNA. And this is a very important type of modification discovered, decades ago, for which now with technology we're able to understand and utilize information a lot better. And the other thing is that DNA is wound. It's not just a linear structure. It's wound, and actually, if you stretch it out from one end to the other, it measures 2 meters long. So obviously, we're dealing with a microscopic molecule, so it has to be very much condensed. And the way that this happens is that DNA wraps around proteins, and it just shrinks it all up. And what happens is one of the ways that we can regulate the expression of these genes is by how loose or tight our DNA is. So again, this is independent of mutation changes or copy number changes, like meaning gains or copies of genes. It's very much dependent on physics in a way, so how loose or how tight the DNA is. So you can just imagine the tighter the DNA, the harder it is for genes to be activated because they're less accessible to various factors that are involved in activating DNA. And so the looser the DNA is, the more accessible expression, the more easily genes are expressed. And so these proteins have all kinds of different modifications. There are about 5 or 6 of them that can alter the function of genes. Okay, and we're trying to understand all of them. We're trying to understand what are the changes that occur, what are the DNA methylation changes that occur. So that's what I mean by methylation. Methylation for any chemist in the group is, organic chemist in the group is a carbon molecule attached, which binds to 3 hydrogen molecules. And that's a chemical modification that's very, very significant to the function of genes. We don't fully understand all the implications, but we're getting there slowly and surely. And so then if you take the DNA again, you wrap it around these core proteins, and these core proteins have these tails which can be modified in numerous ways. So in myeloma, so this is a picture of, this is a picture of what you think naked DNA looks like, which is our prototypic picture that we see everywhere, but it really doesn't really look like that in the cell. It wraps around, and you can just see this unwinding and winding of the structure of DNA. When you look under the microscope, you'll see things like this, which is how our chromosomes are, visualized under the microscope. So DNA methylation, I'm going to focus a little bit on, actually only on that for the rest of the talk. It's a modification that I said involves the addition of a methylation group to a cytosine residue in nucleotide and DNA. It's catalyzed by an enzyme, so there are enzymes that add these methyl groups to DNA. So these islands can cluster together, and we refer to those as islands. And most studies to date have focused on methylation events in these islands, because what happens is, how many people have heard of tumor suppressor genes? All right. So typically we hear about the methylation of a tumor suppressor gene, and that generally inactivates these genes and leads to cancer. But so a lot of the studies until recently with the advent of new technologies have focused on the methylation events that occur in these islands, and they regulate gene expression. Okay, and it's critical for development. So it's very, very much an important process during development. But then, like most cancer processes, things should be quiescent after development for the most part, and then somehow with cancer, some of these developmental processes get reactivated. Okay. So the interest for DNA methylation in myeloma was really sparked for a number of reasons. One, there wasn't that much known about DNA methylation changes in myeloma or any other epigenetic mechanisms, actually. And so I'm sure you've all heard of the Multiple Myeloma Research Foundation Genomics Initiative, which was basically a mammoth effort to characterize in a very high resolution and very fancy ways the compendium of genetic changes, so not epigenetic, but the compendium of genetic changes that happens in myeloma. And this study closed for the most part, and a fancy new paper just got published recently in Nature, publishing some of the next generation sequencing results that were identified in 38 myelomas, a whole genome sequenced for myelomas. It was a very impressive study. And I came into the picture, I actually wish I was part of this study, but I came into the picture a little late. And so with that, we felt, well, the epigenetics piece is missing. B, there isn't that much known about epigenetics in myeloma, so it was an opportunity. The MMR funded a project for us to sort of start embarking on this work. As you can see, this was a multi-institutional effort. So, without boring you with all these details, they said there were very few studies and they were focused only on hypermethylation events. Okay, and there are drugs out there now, epigenetic drugs or epi-drugs that are really being developed. One of which we heard just heard about recently, SAHA, which is an HDAC inhibitor. There are DNA methylating agents in the clinic, azacitidine, and doesn't. I'm very bad at pronouncing drug names, so I'm gonna pass. But there are demethylating agents in the clinic that have shown some efficacy as well. So we wanted to ask the question, very simple question, what are the DNA methylation changes that occur during the progression of multiple myeloma?